Begin now. So, a little bit of housekeeping. I wasn't exactly at the same place with the other group where I left off last time, because I think we had covered some of these protozoans. So, effectively, the last of the significant and the ones that we pay attention to of the eukaryotic microbes are the protozoans because they are very significant. And if I didn't amplify it before, I do wish to. Then what we go on to is this, basically how they grow. So that's the next chapter. So really, and it's funny because they have like an additional chapter. So your textbook, this Cowan, and 3 is bacteria, 4 is eukarya, 6 is the growth. And then when I look over here on the flip side of that, the one 3 and 4 were combined in the Nestor text, which we used for a long time. It's another McGroyle text. And I again, I'm very comfortable with some of the illustrations. So when we do get to this called the bacterial growth curve, and maybe some other illustrations will flip over to that. And these are all, again, available on the PowerPoint. So that's this tiny housekeeping note. And now I just want to go back and complete or reiterate. Protozoa, they play, again, a little bit at the end. Malaria is a protozoan, Trichomonas vaginalis is a protozoan, giardiasis, amoebiasis. These are problems that we see all the time, not just in troubled areas, but in our areas. So we have to deal with these, at least give you the background. So protozoan first animals, again, single cell, very resemblant of us because they don't have any chlorophyll. They don't have a cell wall. There's lots of different species, and they can be and they're land or water-based. Algae is water-based. Fungi are land-based. Protozoans are a little bit of both. And as you can see, the amount of infections and malaria surely predominates from a standpoint of really life-threatening major diseases, major killers, particularly in equatorial Africa, which most statistics will tell you it's the most, versus, to me, the second most common sexually transmitted disease in this country, Trichomonas vaginalis. Fortunately, the trichomonas is a bit easier to treat than the malaria. And that's the big problem. We get to it. They're eukaryotic cells. They're our cells. So if we're going to interfere with them somehow, we're inevitably going to interfere with mechanisms of our own cell. That's the problem. So, yeah, and they have different, I'm not really worried about the plasms or anything. We classify them based on movement. So and again, most happens in the membrane. There are organelles on side. They're relatively tiny. And so we have some larger forms that are there. Their motion can be either amoeboid motion, scooching around just like a macrophage. They can use cilia. They can use flagella. Okay. They ingest their materials the same way we do. They can scavenge. They can even graze on other bacteria or algae. There's little oral openings in some of these, as you will see when we get into the specifics. Some through the membrane. So as you can see, some within the plasma. This is when we start to get into pathogenic or disease causers. Now you're going to begin to see that when we get into those kinds of diseases that are like that within the plasma. That's what we're talking about, malaria. Very, very interesting disease. And moisture plays a significant role, as it does with a lot of microorganisms. The absence of moisture, very few of them can survive in that environment. Okay, and you can see they're done, and they can survive in extremes. I commented a little bit before. The stage that concerns us actively passing it from point A to point B or person A to person B is called the trophozoite. Okay, modal, meaning maneuvering around, mobile, feeding stage, all the things. And unfortunately, and this is one of the things that's pretty hard to destroy when we get into methods of control, cystic forms. So it's not quite an endospore, but it's very difficult to deal with structure. And on sort of the pecking order of how things are easy to kill or hard to kill to obtain sterility, this is pretty high up the pecking order. So protozoan cysts are there. Very resistant, can be easily dispersed, play a role in disease spread. Again, when you're looking at little cystic formations, particularly, it's part, as well as worms, it's what your veterinarian looks at with fecal samples. We have a dog park that has a pool up by us, and they have to, every year that you're going to go, every summer when it's open, they have to have a fecal sample and make sure that none of that is in the dog. Relatively recent on their files or where the dog can't swim. That's called lucky paws in some, or our neck of the woods. So moving on, and to the show, I don't want to go into the life cycle. It goes from basically trophozoite active feeding to cyst, and they can go and do this quite a bit. So we'll get back to those terms down the road. Some are strictly trophozoites, okay? Some alternate. So for instance, Trichomonas vaginalis, they're extraordinarily common sexually transmitted disease. And because it doesn't have a cyst form, it's only when someone actively has this that they will spread it to their partner. And it doesn't matter how it's spread. There's any one of a number of ways to be spread, but most commonly vaginalis. Whereas when we talk about amoebic dysentery or giardiasis, these are particularly contaminated water supplies with animal fecal material. This is if you know somebody who's got a well and there's a problem and it's inevitably close to a septic tank. Big problem. And so we're not that far away from significant rural areas, even though a lot of folks are unfamiliar with it. Most have city water or a water delivery. They don't necessarily have well water. Problem with wells. So depending on where you live or where you work, this may be of significance to you. Typically simple. They may divide multiple fission, like one amoeba into a bunch of giant amoebas, a giant amount of amoebas rather, or very, very asexual mitotic. There is some sexual, but we won't delve into it. And this just gives you some of the ideas. The amoebic dysentery, okay, Giardia, and those are big players around here. Trichomonas. This is typically in fouled areas where animals, and we are going to see this, we have a lot of these problems are where animals contaminate water supply and people have to use them. So we don't see it around here. But if you're in, again, Africa, India, wherever you are, the water supply, leishmaniasis, another one. Typically it comes from urine and water that people bathe in if you're bathing in a very rural or underdeveloped area, just to give you. And then these are all the protozoans, the plasmodium, particularly vivax, falciparum, and malaria, all human and vector-borne. They infect us. Toxoplasmosis, you in nursing will hear plenty about that. That's the whole one that we see in the cat litter associated with cat. That's why I always tell them, at one point we had 8 cats. 8. So we had 2 dogs, 8 cats at one point in one house, plus other animals. And so imagine we had like 3 litters that were changed constantly. And we were still trying to have children biologically. Again, clarifier, my 3 children are adopted. So I didn't have biological children. My first wife and I, you have to change all the kitty litter. So I think that was the reason, well past childbearing years. Moving on. So you'll hear about that and some of these others you'll become acquainted with as well. And we don't do the worms. You want to look at it, it's kind of scary. Ugh. I mean, it's much more of a problem vis-a-vis this. And around here, very significant in animals you have outside, because now you're dealing with heartworm, pinworm, you're dealing with Lyme disease. So flea and tick collars, making sure that they get regular veterinary checkups, all of those things. Use whatever means you feel appropriate, but please do so. And that concludes our little nugget on sort of the basic of eukaryotic microbes. So our next unit, and the last unit that's on the exam, is this forthcoming unit, which has to do with growth in general. So growth is so you'll find it in chapter 6. So a little less on the nutrition, more so on the growth, because we're really dealing with the ones that we come in contact with most of the time. And without going crazy about it, again, the essential. It's just like you learned about in A&P1, essential amino acids. Essential biologically in a nutrient means you have to acquire it through your diet. You can't get it. Your body, we have 8 or 9 that are essential. We have to get through our diet. Everything else, we can take those and make them. Okay. So when you hear about a complete protein, animal proteins, our fish proteins, are all milk, which is effectively an animal protein. As long as you have that, you have a complete protein. People who are impacted by it are people who don't consume animal products, vegans, and then you have this magical combination of legumes and seeds or nuts. So that's where beans and rice come. If you did not know, the only legume that's really a complete protein that we consume a lot of is tofu, which is why tofu is very, very popular. We have tofu dishes all the time, even not being vegan. I have my stepson's vegan. So macronutrients, the ones we need a lot of. The big players, when it comes to us, and it differs between us and bacteria, let's say, carbon, hydrogen, oxygen, and nitrogen, not necessarily water. And then typically what we use a lot of after that is calcium and phosphorus. Okay, think of bones, which are basically calcium and phosphorus, so we need a lot of them. Yeah, so that's for us. So let's say that's for human. If you look at bacteria, you have carbon, hydrogen, oxygen, and nitrogen. Same before, phosphorus, but sulfur. Sulfur plays a bigger role than calcium does. Not that calcium is not, it's considered a macronutrient, but not in the same category. So if you think of the top 6, they're different from our needs, particularly vis-a-vis bones and a lot of our maintenance systems, as many of you have or will see in AP2, are calcium based. Blood clotting based in calcium, lots of stuff. Calcium plays a triggering role in a lot of things. So micronutrients, sometimes called trace elements. So when you take a look at a package of something you're consuming, let's say a pack of vitamins, when you look at the vitamins part of it, many of them are in small quantities. When we look at the minerals, typically that's where you see this. Certain amount of this, certain amount of that, zinc, nickel, etc. And most of the time, if you eat a balanced diet or if you take a multivitamin, you got it covered. Okay? So that's the distinction between. And some of them are really important because they play a pivotal role in proteins and making sure the proteins kind of form around that. The classic one that you may or may not have learned about, hemoglobin, which is around iron. Okay? So we have to have a certain amount of iron in your diet. That's there. So, and basically the distinction they talk about, and this is where they come from, and I never do it, and I'm not going to. Organic means that it's carbon, basically carbons, oxygen, and likely some hydrogen, and covalently bonded carbon. It's almost a watchword from that, along with carbon and hydrogen bonds. I mean, CO2 is inorganic by definition. Even though it's got a covalent bond, it's the oxygen. There's no hydrogen in the party. Okay. So some of that, I emphasize that when I do AP1. Okay, I don't. Whoever was your instructor, it's up their determination. Just like us, bunch of water, proteins. Here's the chomps. Most chemicals available as compounds, not pure elements. In other words, most time they break them down just like we do. And again, we have a variety. And so they go through all this stuff, and I'm not going to do the weights. And don't trouble yourself with all this. But that's an E. coli. Great. So you can see, I mean, that's where they come up with sulfur. It doesn't minimize calcium or potassium or sodium. They're just a little farther down the picking order. How do we classify? Touched on it just a smidge before. We are, by definitions, heterotrophs. So the organisms we are concerned about are called typically heterotrophs or chemoheterotrophs. So, and it's all based on how do you get your carbon products and where do you get your energy. We get them both from organic compounds. So in the global scheme of things, we're called organochemoheterotrophs. We eat a lot of different stuff that are in the family of organic chemicals, and we get both our carbon that we can make other stuff with and the energy by breaking apart those wonderful covalent bonds, and voila, that's us. Autotroph, okay, self-growth. So that's a plant or a photosynthetic bacteria or algae. It uses inorganic carbon dioxide, and as I was talking to you before, it fixes the carbon, meaning staples it together, okay, to make carbon compounds. It's also called a photoautotroph because its energy comes from the sun. So it requires light energy, some kind of a photo, depending on its environment. And so typically light energy is its energy source, and atmospheric carbon and atmospheric and water within the environment are the appropriate amounts. So it converts carbon dioxide to organic compounds, and it's not dependent like we are on other things. Whatever you want to define as living things. So a vegan, they're still eating plants. There's again no PETP. There's nobody for the people for the ethical treatment of plants. Animals, sure. Bacteria, no. Plants, no. You just ate that broccoli. You're a murderer. It was living at one point. I expect everybody to have signs out here tomorrow in a protest movement. Unfair to plants. We eat them. I like Ivy. Whatever you. There's a slogan called "I like Ike" for President Dwight David Eisenhower, who was the president before John F. Kennedy in roughly 52 to 60, 2 terms, and that was the big slogan was "I like Ike," which is very catchy. And there's other ones. So microbes that photosynthesis, chemotrophs, microbes from chemicals. So there's variations. The big players are this: photoautotrophs, typical plants, algae, pigmented bacteria. Oddballs, don't have to know. Methanogens, rock eaters. Doesn't sound very tasty. I had rocks today. Oh, delightful. So did I. So these are oddballs. That to give you an idea that microorganisms and that's when you start looking at some of those archaeal organisms in extreme environments. And look, they can't go out to McDonald's. There's no Happy Meal for an archaea in the middle of a volcanic caldera. No one's delivering. I got to incorporate that. That's a pretty good thing I just came up with. Versus, and again, photoheterotrophs, oddball, but again, the big ones that we see, this chemoheterotrophs, that's really what we are, and we even classify as organic. All you got to know. No. Parasites. So the problem with parasites is just right here. It basically is gobbling up whoever it's occupying, be you, a bacteria, a plant, an animal, whatever, from a living host, and destroying it in the process, damage or even death. And all sorts, and they cover all types of things as we will cover them. Whether they live on, ecto, outside of, or inside, different thing. We'll talk a lot about viruses, which are classified as intracellular parasites. So the term would be reside rather than live, because remember, they're dead. They're not live or dead because they don't fall into that category. So, I mean, a little bit things like that. And so some of these are really unusual. So here's the one that's classic, bacteria called syphilis, which we've never been able to grow outside. And indeed, the bacillus that causes leprosy is also very, very difficult to cultivate. Okay, and so there's a lot of this I would classify as background material as of this, and you can scroll through it if you're interested. And you probably had a very similar unit in anatomy and physiology where you could scroll through a chart and see what iron does and cytochrome proteins, and zinc, genetic mechanisms, etc. Okay, so as again, transport across the cell membrane. Again, movement of molecules or atoms, and you need to review this. Again, this is part of what are called prerequisites for any course, whether it's a government course or sociology or psychology. When you talk about here, diffusion, facilitated diffusion, osmosis, active transport, all those things are here. So diffusion, you don't, any of these that don't have to expend energy are used because you don't have to expend energy. And energy resources are precious for you and I or for a bacteria. So the molecular movement and be aware what drives the bus, the most important factor, no matter what anybody says, concentration gradient. More here than here. Okay? If everybody's concentrated sitting over there and all the seats are taken, you're going over here. When things are concentrated, the molecules or the entities bump against each other, so they're going to go find sites where there's more space to go to. That's the whole principle of concentration gradient. That's the player. You know, we can talk about size, we can talk about weight, we can talk about charge, but that's the big thing. So anything in a living system like we're describing, we use the term solvent for water, because everything is dissolved in water. Okay, it's a solution. So whatever it is dissolved in is called the solvent. Whatever is dissolved in it is called solute. Doesn't matter what it is. It can be a protein, it can be calcium, it can be hydrogen, glucose, doesn't matter. Okay, so water follows the same guidelines. This is osmosis. And I'm sure you looked at it ad nauseum in A and P. Basically, water will go from a higher, just like anything else, higher concentration to lower concentration. So it will flood cells, and most cells are permeable to water. That's a big problem. Okay, so our cells, if we are overhydrated, our cells burst. We don't have the luxury of a cell wall. Plants and bacteria do. So I'm just going to drink a ton of water for my GI infection. Probably not going to help you. It'll probably help you rehydrate, okay, if you had diarrhea and vomiting, but it's not necessarily going to help you get rid of the infection. They're going to be able to withstand it. Need something else. And so that's the whole idea. What you will see universally as when we remove water by inducing things that pull water out, whether it's salts or sugars or other materials, they'll pull water toward them, and the bacterial membrane will collapse and pull away from the cell wall. And remember what I said, it's an envelope. It will be destroyed. That's why the salting and sugaring is something we call in the food science curing. Okay. I don't know how the name evolved, and I usually like that type of stuff. So here, and they're just kind of showing you a little bit about that. Classic experiments, okay? And maybe you did it in your lab. I used to do it in my A&P1 labs when I haven't had it at the school a lot. I usually don't focus on it there because you've done it previously. So here's a bacteria, for instance. So if you put it in an isotonic, in other words, same relationship, solute to solvent as anywhere else, as the bacterial cytoplasm, as its normal environment, the same way with us. Same, that's inside our cells and outside our cells. Whatever the abundance is, it has no effect. Stuff will move in and out and basically come toward what we call an equilibrium. You know, you looked at that, sodium moving in and potassium moving out, all that jazz. Okay. Hypotonic. It's got a cell wall. It'll swell, but the cell wall holds it in place. Remember, happy plant? Come on, water me, I dare you. Cool. Versus this, it'll shrivel. And that's what I meant. So we call this plasmolysis. It causes lysis, breaking of the plasma membrane. Different, and let's say if this was a typical study, it would cause it to burst. No cell has normal, no effect. Enlarge and burst. Okay, what happens? We see this when we add it to blood. That's a big problem. Overhydration kills somebody who has can pulling water out. The fancy name for it is called crenation. Crenation. And you may have done that with like aseptic sheep blood cells, put it with salt water, and you look at it microscopically, it starts to get, it's no longer a disc, it starts to get little squared ends. That's kind of a classic experiment. And facilitated, same thing, passive, concentration gradient. What facilitates it? A protein passageway. We call a gate of some type. Okay, so a transport protein. So the difference is we'll either have transport proteins or sometimes we'll have channel proteins. The implication of the word channel is like it's always open. When you have a transport protein, the implication it can open and close based on something contacting it or environmental factors, etc. Overview stuff. And everything else is active transport. If you're going against the grain, if you're trying to, class just was over. The other class been waiting to get in to have a test. They're kind of coming, you're pushing your way out. That's going against the grain. You know, go to, if you ever go to like a Steeler game with 70,000 people, everybody tries to get up before halftime to run to the bathroom before you can't even get in. And all of a sudden you're coming back and everybody else is going against you. It's like, and you get called a lot of unpleasant names. They question your parentage. They remind you to do an anatomically impossible act with yourself or a farm animal of your choosing. You haven't heard that one before.
No.
See, the rest of you are looking at me like, we heard that last year. Right, Michaela? So active transport, very specific, requires energy, used a lot for and again, we limit this. Whenever all that exocytosis, endocytosis stuff you learned about, anytime you're making little vesicles, which we don't do in bacteria because they don't have them, okay, but we do in our cells, energy dependent. So all of those cytoses are making membrane and opening and closing it. That requires energy. It's like group transport. Get on the bus, we're moving. Okay, voila. Bingo. Finally, something relevant. Okay, that's the background stuff. Temperature. To say in my old neighborhood. So, temperature is interesting. You're going to find a specific control the growth of microorganisms. It's temperature. What do we do when we buy something in the store? We're not going to use it for a couple of days. Put it in the fridge. Put it in the freezer. Right? What are we going to do once we've done that and it's a raw protein? Fish. I mean, other than, unless you're in touch with your inner sushi and sashimi, it has to be sushi with sashimi grade. What are you going to do? You're going to cook it. Right? So you're denaturing the protein. Why do we use heat? Okay? When you think about it, we look at growth versus temperature. Okay, as we increase from lower to higher. Now, normally when we look at living systems with their growth, which is depending on the factor, usually it looks like this. You've all seen this. A bell curve, right? Remember bell curves, Gaussian distribution, normal distribution, middle, mean, standard deviation, type of statistical analysis. Not temperature. Okay, you start with a minimum. Basically, when bacteria grow, they start this way. As you increase the temperature, it goes up looking like a bell curve, and then boom, falls off the top. So what that means is your minimum temperature, your lethal temperature, and your optimal temperature, the best temperature for you to function at, temperature optimal and temperature of death are close to each other. What's our normal in Celsius? What's our normal in Fahrenheit? Well, 98.6, right? 37 degrees Celsius. So each degree of Celsius is equal to 1.8 degrees Fahrenheit. So if you got 104 fever, okay? So now you're at, if you do the math, you went from 98.6 to 104, 37 degrees Celsius, 40 degrees Celsius. You're really sick, okay? You go up another 2, at 107.6, most likely you're gonna die. So 37 to 42, 5. That's the idea. So temperature is a reliable way to destroy things, and we have to be cognizant of that based on the environment. So we do a classification. 3 of these are relevant to us because we either are in them normally, the one is obviously around 30 Celsius, or ones that are close to it when it's considerably warmer. My wife and I will go freeze over. Usually go in the summer, it's like 100. The whole desert thing. So you have minimum, maximum, or optimum, okay, et cetera, et cetera. So they use this. So let me show you I'd rather show you the Nestor classifications. I think it's visibly more or graphically better. Here they are. So here, I'll show you the names and I'll show you, and they talk about temperature, and I'll go back to the classifications. Okay, this is the temperature graph. So, and it's probably a little bit better. The ones that are significant to us are here. Nearly all of the microbes that hurt us are meso. Meso means middle. They like the middle, mesophiles. Typically, and there's overlap, as you can see. Look, that's certainly not a bell curve. It's a long cliff. 25 to 45 degrees Celsius. Normal range. Most pathogens very, very close. And that's interesting. Leprosy. Why does leprosy target extremities, fingers, toes, noses? Because it's colder. We call those acral, A-C-R-A-L, parts of the body. They are, if you do skin temperature readings, which are easy to do with a sensor, those are the coolest areas, and leprosy goes better away, lower than normal. That's one. I mean, it's easy to understand how disease works. So you can see it. So 37 is pretty darn close to death temperature. Like I said, with us, 42 degrees circle. Psychrotrophs. Okay, you can see there's a certain amount of overlap with mesophiles, roughly 15 degrees. Your refrigerator is probably a tad colder than that. Probably a better range to say 5 to 30 degrees, I would think, than that PowerPoint. Typically, your refrigerator is about 4 or 5 degrees above freezing for the most part. Newer refrigerators will show you, and you can obviously set it if you want. And what does a refrigerator do? Does it kill anything? No. It just slows it down. Cold slows down the metabolism of us, and it slows down the metabolism of microorganisms. So the beauty of cold is just that. So theoretically, when it goes up back to temperature, everything will be fine. The reality is there are organisms that still work in the cold, and so you have let's call it a limited refrigerator shelf life. Like you can't leave and put a thing in milk, even if you close it. They come back 3 weeks later. Most likely that milk is already spoiled because there's spoilage organisms in there. They're still living. They're just working a lot slower, but eventually they'll spoil it. Thermophile. They like it hot. Light increases in temperature. Well, again, there's a certain amount of overlap. They probably start around 40 to 45 degrees Celsius. And they really, and 70 is pretty darn hot. We're not going to have an environment here. Where would you find an environment like that? Go to a hot springs, or as many people like to do, go to a hot tub. There's a lot. If you have a hot tub, we have one sunken into the deck one side. Everybody loves to go to those things in the winter too. Just can't walk outside when the snow's on the ground. And all those clothes on. When you go in there, it's like margarita, mojitos, midnight. Just don't. I'm a lot. Dr. Dress, used to be our department chair. You can throw things. Your bag. Yeah. So those are the big three to remember. Hyperthermophiles, usually archaea, like vents, by a geyser someplace in Yellowstone, whatever you want. And then psychro. Psychro is not psycho, it's psychro means cold. Hyperphiles, again, polar ice cap type of stuff. That's why temperature is so important. So the big three are here, but you should be familiar with all of those. And really the take home point for all of this is very much, uh, very much about the effects of temperature. So I'm going to go back and head back to that other PowerPoint that we were using before. And, this is the one that they had. So they're looking, and they have different temperature levels. Obligate can't grow above 20 degrees. We'll see them more in the freezer. So they actually can grow in a refrigerator because they could even grow at freezing. Okay. Fortunately, they don't present a lot of problems to us. Psychrophiles can't grow above it, but cause them to grow rather than inhibiting them sometimes. It depends. So that's the psychotropes, the one we're talking about. Have optimum temperature, roughly there. Again, dairy products, as you'll find out, have a bad tendency to have staphylococcal. When you get food poisoning, typically it's staphylococcal food poisoning from dairy products. And if you pay attention to the news, this you do not want to have. Because you'll see it all the time. Just Google Listeria monocytogenes and look at an outbreak and see what this is. For immunosuppressed individual, for pregnant women, it is a big problem. And you get that with food contamination. I remember there was a case where they were it's like if they got cantaloupes, you pick them off the ground. Now, I don't know, when you go to the store and pick up your cantaloupe, most people don't go home and they just take a sharp knife and boom, right? Wash the cantaloupe. Scrub the outside. You scrub your cantaloupe. Who does it? Get that spritzy thing, because whatever that knife passes through on the rind goes into the flesh, which ordinarily would not have anything into it. And then you consume it. That was a big outbreak that killed a few people. Okay. Mesophiles, most of the time, all over the place. Okay. Thermoduric can live with it. So, for instance, this is where those endospore formers, they call them thermoduric. Again, boiling them is not going to do it. We'll get to that. So they like it hotter, compost piles, a lot of variation. That's there. This is their chart. I didn't like nearly as much. Oxygen, and then we look at the oxygen levels. They do in this, they do the growth curve a little bit differently. Okay. So oxygen is a little bit different, and there's 5 categories that are there. So it has 2 effects. Sometimes like you and I, we have to have it to survive. On the other hand, and this is what I was telling you about when we talked a little bit about the superoxide dismutation catalase, oxygen produces toxic derivatives. And so even bacteria have to have mechanisms if they're exposed. You don't have to use the oxygen, but if the oxygen is in their environment, they have to be able to deal with it. And that's what this is. So the categories, they use oxygen, that's what we do. Okay. They can neither use oxygen nor detoxify it. That's what we call a strict or absolute or obligate. The term we use scientific is obligate. It means it's obligated not to use oxygen. Oxygen kills it. And some can use the oxygen. Okay, they don't necessarily use it, rather, but they can deal with it. And you're going to see that. So the oxygen, what they call singlet oxygen, In other words, not O2, but just an O with some electrons floating around. The one I showed you before, superoxide, the nastiest. Hydrogen peroxide, we use it a lot as both an antiseptic and a disinfectant. We'll get to why. It's something called hydroxyl. So without going to the chemistry part of the class, they're all toxic derivatives of oxygen. Again, an oxygen environment or one that uses oxygen like we do have to have a way to deal with these bad things. And this is the chemistry. Don't remember it. This is what I was drawing on the lab. Superoxide dismutase with some hydrogen ions. It makes it peroxide, gives off oxygen, and then the catalase does that. It rapidly converts it to water and oxygen gas, which is great. Okay, so I'll show you the categories on the other PowerPoint. Again, because I'm used to it, I don't want to confuse you. Oops, wrong guy. Here they are. We never did this experiment here. I've done it at other schools I have taught at. And you'd get a tube. Really? We had to do that out of nowhere? What did I do to deserve that? Are you kidding me? So what happened with these kinds of tubes, you have a special agar in the tube that would solidify, not all liquid, warm it up, you'd inoculate it, swirl it around, let it grow, seal the tube. And what would happen at the top of the tube, and you'll see this yourselves, you'll get aerobic things that'll crust on the top. And stuff that only grow at the bottom of the tube are mostly anaerobic. And because of this positioning, we have 5 major categories. What a lot of bacteria are, like that exist, let's say, in certain environments, obligate aerobes. They have to be oxygen. They can't survive without it. Obligate anaerobes, just the opposite. That's botulism. That's tetanus. They only can grow and multiply in an anaerobic. Why C. diff is such a poor problem? It exists in the colon, and the colon is an anaerobic environment. So you're going to see some oddballs with it. So interestingly, when you're in class, in the lab, and growing E. coli, whether on a plate or in a tube, it's called a facultative aerobe. It can use oxygen, but it doesn't require it. It has the faculty, meaning capability. In the term of faculty here, it's different than a faculty member like I am at a university or anywhere. Okay. Facultative means it has the faculty or the capability to use oxygen, but can do so without it. And if anything, they like to ferment because it's relatively easy to do. Some require only a small amount of oxygen, but they do have to have some. And some have nothing to do with oxygen. They can grow in it, but they don't use it. So we refer to them as aerotolerant. They can tolerate the air, but they are obligate fermenters. The only way they can do energy transformation. We're talking bacteria. They're not us. Okay. So they're different in that idea. So the notion that you have to have an electron transport chain or a Krebs cycle does not apply. Okay? You and I, if we start to ferment, we're dying. If your heart stops, you start to ferment. And if someone goes into a code, one of the things we usually shoot into them is a couple of units of bicarb because we're trying to neutralize all the acidity that's being produced. It's not going to magically make them better, but if we are able to resuscitate them, at least the high degree of acidity will not denature a lot of vital proteins and therefore vital systems. That's the idea. So this is what it looks like. We'll finish with this. They grow when oxygen is available, typically at the top of that tube. Use of oxygen required. They have superoxide dismutase and catalase. Gotta have it, just like us. That's us. Grow best in the presence of oxygen. Facultative. Facultative is an important word. What we work with in the lab are mostly facultative bacteria. They're easy to grow. That's what we use. Good news. Bad news, hard to kill. Because if it's easy to grow, it's hard to kill. We deal with what are called, it's a fancy word that I'm sure we'll hit again, called fastidious. Fastidious are hard to grow, and that means relatively easy to kill. Because all you have, all you have to do is typically put a whiff with an appropriate antibiotic near them, and they're done. So, facultative, that's when oxygen available, but you go without it. Uses it if available, and it produces the same two enzymes to deal with the oxygen. Obligate anaerobes, they're at the bottom of the tube, they're throughout. Can't grow in the presence of oxygen, don't use it, does not have superoxide dismutation. That's why when we look at a wound, we know whether it's going to be tetanus or non-tetanus wound, depending. And typically, the only wounds you worry about are the ones that become infected, or the ones that have a lot of dead tissue with it. A war wound, somebody gets in the backyard, tears, and regularly does things. So we do a whole wound. Microaerophiles, only if small amounts of oxygen are available, between a roughly oxygen environment of 2 to 10%. Ours is either half to as little as 10% of our oxygen. They are significant. We'll get to there's a couple of diseases that are very significant. Flesh-eating bacteria, microaerophiles, very interesting. Strep pyogenes, which is strep throat, strep are unusual. And the other one, we'll look at a little bit, Helicobacter pylori, the one that's in the gut association to some extent with gastric ulcers. They are aerophiles. They need oxygen for a ton, because they have some. And here, we don't even have the catalyst. So it only produces superoxide dismutase rather, doesn't use the oxygen. It doesn't really matter whether oxygen is present or not, but it has at least 8 meters. And those are the oxygen-based categories. The most important part of this chapter we will do Monday, which is the bacterial growth curve, but we will not get to it today. Enjoy. Thank you very much. Have a great weekend.